Okay, so today we are going to do the light reactions for photosynthesis. Now, we talked last time that during the light reactions, we are going to form two different things, and that's ATP and NADPH. And if you remember, ATP, that's adenosine triphosphate. NADPH is going to be the molecule that we are going to use to pretty much transfer electrons. It might be holding an electron at one time. The next time, it might be giving that electron off. So we're going to go through some colors that deal with photosynthesis. Just so you know, we're going to talk about wavelength. All that is going to be our distance between crests of waves, right? Nothing too crazy there, but we're gonna mention them often, and there's going to be some different numbers that are associated with different colors with respect to wavelength. So we're going to be using the visible light spectrum. I know there is a bunch of different areas of the whole spectrum, whether it's microwaves, radio waves, x-rays, all these different things. We are going to be focused on the part that we can see, which is just, visible light. So with this, we are going to be using something called photons. And we talked about like protons, neutrons, electrons before, and they all had these very small masses to them. Photons, these are going to be the particles that make up light, okay? So just fancy name for light particles. Here is our visible light spectrum, and we have the wavelengths down below. Now, if it is a shorter wavelength, okay, which is going to be not a whole lot of distance between our crests, we are going to have a higher energy. So purple, blue going to green, these are going to be our high energy colors. On the other side, we're going to have a longer wavelength. So the red, the oranges, these are going to have lower energy on our visible light spectrum. Okay. All right. So as the plant is going to absorb or reflect these different colors, okay, we are going to have different wavelengths and different energies associated with them. Obviously, green is not going to play a big role here because the plant is green. It's actually not absorbing any green. It's reflecting all the green. But every other color is going to have a different wavelength for our plant. And you guys can see here, this is just our green light being reflected and all the other colors being absorbed. Now, what are they being absorbed by? If you guys see, we have the thylakoid there. Inside of our thylakoid is the chlorophyll, all right? And that's what's going to absorb all of our sunlight. This just goes over a device, guys. It's called a spectrophotometer, all right? All this is we're going to look at the pigment, and this is going to tell us which wavelengths it's going to absorb. So pretty much, guys, that'll tell us what colors it's going to absorb and which ones it's going to actually absorb the best. All right. Here's just the example, guys. I'm not going to go over that with you. Now, we are going to have two different chlorophylls that we're going to talk about. We're going to talk about chlorophyll A, and we're going to talk about chlorophyll B. Chlorophyll A is going to be more absorbing blue light to violet light. All right. Chlorophyll B is going to be pretty much focused on red light. And if you see there, it says carotenoids. Right? Pigments of carotenoids, it actually is in carrots. Right? That's one of the pigments in there. But chlorophyll A, once that one goes away, we get to see all these different colors, all these carotenoids in the fall. And that's giving us the different colors of the leaves in the fall. So that's all we see after the chlorophyll goes away. Here is a graph of our wavelengths here. You guys can see we have chlorophyll A, which is like that teal color up there, and it tells us the wavelengths it is going to absorb best. So chlorophyll A, yeah, we're in that purple-blue area, and then if you look down the road here, it's also absorbing the red pretty well. Chlorophyll B, same thing. All right, we got the purple-blue area there, and then we have a little hill on this orange area here. Okay. So what color do you think would be best to give the plant? obviously white light is going to be the best because white light is going to give us every single color in the rainbow. But let's say we don't have white light for some reason. Well, if you could see here, it's absorbing blue really well, and it's absorbing red really well. What color has blue and red in it? Purple. Mix blue and red together, you get purple. So if you don't have a white light to put on a plant, purple would be the best, next best option. Okay. All right. Okay, so for our light reactions, we are mainly going to deal with chlorophyll A. Chlorophyll B, a little bit, but not so much as chlorophyll A. So that's going to be the main one. And we already talked about the carotenoids. All right, they're going to absorb a little light, but not too much. Good there. Okay, we actually already talked about this one a long time ago, guys, when electrons get excited. You remember when we talked about them, I think it was chapter 2. They can go to different energy shells, different states. In other words, absorb energy and go further away from the nucleus, okay? That's all this is talking about here. And you guys can see here that once the electrons have an excited state, they're going away from the chlorophyll. And I'm going to show you that in the diagram I'm going to draw here shortly. So we're going to go over two different photosystems. Now, we talked about yesterday just a little bit our light reactions and then our Kelvin cycle. So we're going to start off with the light reactions, and the light reactions is going to be composed of photosystems. And all this is different systems that are going to absorb light, and in absorbing that light, things are going to happen, all right? And we'll go through all of them right now. So I'm going to kind of make this fairly messy at first, guys, but don't worry, I will clean it up. Let me draw our chloroplast over here, and I'm going to do it relatively small. So two membranes, we have an outer membrane, we have an inner membrane. Then we're going to have these thylakoids here. Okay. All right. So we have our thylakoids. This area out here would be our stroma. Okay. And that's pretty much like the cytoplasm of our chloroplast. Okay. So what I'm going to do here is I'm going to take this little area right here. If you could see, I got a little bit of my thylakoid membrane in there. And what I'm going to do is I'm going to bring it over here, and I'm going to draw that area. Okay? I'm going to use different colors. I'm not just going to use green. And I'm going to try to make this as neat as possible for you guys. All right. All right. So on our thylakoids, we do not want things just passing in and passing out very easily. So what the thylakoid membrane is going to be made out of is a bunch of phospholipids. Okay? Okay? There you go. And that's going to make sure, things just don't pass through. We're going to be selectively permeable about what can pass through into and out of our thylakoids. Now, let me label some areas here. So if you look here, I took out this little part. So the part on top, this would be our stroma. And then our part down here, this would be inside the thylakoid. Whoa, I did not mean to do that. Sorry, guys. Let's minimize me back down there. Inside our thylakoid. Okay. So, here we go. What we're going to do, guys, is we're going to start off. We got our membrane here. What is inside? Let's start off there. What is inside of our thylakoid? Well, in this area down here, we're going to have our chlorophyll. And that's going to be throughout the whole process, okay? So the first thing we're going to do is we're going to use that chlorophyll, and we are going to absorb some sunlight, okay? And that's going to be absorbed by this thing here. Now, here's the weird part to the photosystems, guys, okay? We are going to be composing this of two different photosystems. It's going to be Photosystem 1 and Photosystem 2. Now, naturally, logically speaking, we are going to start with, yeah, you guessed it, Photosystem 2. I did not say that incorrectly. We are literally starting with, and I will label it up here, Photosystem 2. Okay? Why we're calling it Photosystem 2 is because this one was discovered, this part of the photosystem was discovered second. Okay? Photosystem 1, which happened second, was discovered first, and they gave it the name Photosystem 1. They thought Photosystem 2 was going to happen after, but in actuality, it happens before. So instead of changing it, let's just make it difficult on everyone, right? All right. So this is going to be Photosystem 2. So what we're going to have is our photosystem here. Okay. We are going to have some chlorophyll in this area right here, and it's going to absorb the sunlight. Now, the wavelength of this area is going to be P680. What P680 means is that our photosystem 2 is going to deal with chlorophyll A and chlorophyll B. Okay, they are both absorbed really well. They work really well at a wavelength of P680. Okay. What we are going to do is once this absorbs this light, we are also going to bring in, if you remember our photosynthesis reaction, we have water, we have carbon dioxide, we have sunlight, and then we are going to get glucose. We're going to give off oxygen, and we're also going to get some water out of it. Now, we are going to start off with the sunlight in the photosystems. We're also going to start off with the water. And what's going to happen to the water is it's going to get broken down into some H pluses and some O2. Now I know you guys are thinking, okay, come on, you got an O here, now you got two O's over here. Yeah, we're going to need to split more than one water molecule here, okay? So what happens here is we get H pluses and we get some oxygen off of it. Where does this oxygen go? Oh, that was a bad square there. Let's try that again. Where does this oxygen go? The oxygen gets released and leaves through the stomata. Cool. All right. What's going to happen here, guys, is we have these H pluses, which are essentially just protons. We are also going to get some electrons out of this. Okay. What's going to happen here is the electrons are going to go through our photosystem, through photosystem 2. And they are going to, because of the sunlight here, they're going to get excited, which is why they're going up. They're, oh, look, I got all this energy. I'm getting excited. Wonderful. Okay. After this, our electron, which is now excited, is going to go through some cytochromes. Kind of like the same thing we talked about last chapter, guys, with our cytochromes. All right, we're going to have 3 of them. Really don't have to know the names here. We got PA, CYT, okay, and then PC. our politically correct cytochrome there. PA stands for plastoquinone. We have cytochrome T here. Okay, it's like B6F, I think is the name of it. And then last one, PC, yeah, it's not politically correct. It's plastocyanin. All right, it's a copper-containing cytochrome. But anyway, so all they're going to do is help to transfer this energy. So this electron, all right, which is now in the stroma, is going to go through, and just like before, it's all excited. These energy cytochromes do not want that excited state, so we're going to take that electron and we are going to bump it back down into the area inside the thylakoid. All right. In this process, I'm going to have to, like I said, guys, it's going to get messy. I'm going to have to erase this now. All right. So we still have some phospholipids in here. All right, good enough. Okay. And I'm just going to make 2 more here. Okay. So in this process, guys, we also are going to have some H's here. All right. I'm going to go and do photosystem 1. So I'm going to draw a little line here going down the middle. I'm going to do photosystem 1, but we're going to worry about these H pluses in a little bit here. So that's photosystem 2. All that happens, we split water, we bring in sunlight, we get this electron, it gets excited, it goes through the cytochromes, and now we're here at the end of photosystem number 1. Okay. Now we're going to move to photosystem number 2. We're going to have some more chlorophyll here, but different wavelength. It's P700. So this is photosystem number 1. Instead of having chlorophyll A and B, this one is only going to be mostly chlorophyll A at P700. So only chlorophyll A is going to work there. Okay. Just draw a few phospholipids in there. I'm not going to go crazy with it because I'm going to have to draw some more stuff downstream in a second. All right. Let's bring in another cytochrome here. I'm just going to bring in 1. Okay, we're not going to worry about the name of it. But same thing happens here, guys. We are going to once again have this electron. We got the chlorophyll, it absorbs the sunlight, so it's going to get excited once again. That electron's going to come up here, go through our cytochrome. All right, it's not going to want to have that electron, so it's going to get rid of it. Okay. Now this electron, once it goes through this thing here, which is it's an enzyme. Okay, this guy. I'm sorry, not yet. We got to draw the enzyme here. So our cytochrome is going to get rid of it. Right. We are then going to have an enzyme right here. Right. This enzyme is called Nadp Reductase, ASE enzyme. Okay. The electron is going to go there, and then that electron is going to get used. It's going to get paired with NADP plus, and it's going to reduce it to get NADPH. Okay? And that is the end of photosystem number 2. Okay? I'm not going to go crazy with drawing all these phospholipids in, guys, just to save time. All right. Let me just draw the ending here. Good enough. So now's where the big thing comes in here. So we are going to bring in one more guy. We'll make them red. Yeah, we'll make them blue. Okay. So one more guy here. And you're going to recognize this character. Maybe not by my not so fantastic drawing, but you will remember him. Okay. So this right here is going to be ATP Synthase. Now this is going to be, it's not in photosystem number 1, okay? What we're going to do is we're going to bring these protons, these H pluses over here, all right? And we are going to feed them through our little chamber here on the side of ATP synthase. So now we're going to have the proton on the outside. In feeding them through, our little drive here, our little motor, just like in cellular respiration during the electron transport chain, is going to start spinning, creating potential energy. And in that, erase this here so we have a little more room. With that, we are going to be able to bring in ADP and a phosphate, add that proton in, and we will get ATP out of it. Okay? This is the end of our light reactions. So we have Photosystem 2 where we start, just to review everything, guys. We have Photosystem 2. During Photosystem 2, which happens first, we bring in sunlight and we split water. The electron becomes excited. The electron is then going to go to photosystem number 1. That electron, which gets excited, again in photosystem number 1, is going to go to an enzyme called Nadp Reductase. That is going to reduce NADP plus to form NADPH. So, photosystem number 1 forms NADPH. That's all that is formed in photosystem 1. That's our main goal. Photosystem number 2, it's going to bring in light. It's going to split water. So when is water split? Photosystem 2. When is light absorbed? Photosystem 2. When is NADPH formed? Photosystem 1. The last one we got to talk about is ATP. ATP is technically formed by the products of photosystem 2. Because once water's split, those protons are going to go, they're going to go to ATP synthase, and then that's going to help us generate the ATP. Okay? So that is all of our light reactions, guys. Yeah, that was a lot, right? Now, NADPH, we just get a little bit of it. ATP, we just get a little bit of it. We do not get enough ATP to keep the cell going. That's not enough energy. This is why we still have a need for the mitochondria. Okay, this is everything we just went over, guys, with the photosystems. All right, I like to draw a picture and explain the picture. I think it works better. And this is everything we just went through. The cytochromes, everything like that. Okay, so that's why I'm just skipping all over this. If you would like to read it, you can. All right. You guys can see your book says Photosystem 2 is the one that makes ATP, even though ATP is made downstream of Photosystem 2. It's still kind of, the products of Photosystem 2 are used to generate ATP. Okay, so let's stop here today, guys, and then tomorrow we'll do the Kelvin cycle. Have a good day.